So good morning and welcome to this second day of the IMA Veterinary Awareness Days meeting and welcome to the breakout session on animal welfare. So I will moderate this session and my name is Raffaella Corvi. Just to introduce myself, I work for the European Commission Joint Research Center, which also hosts the European Reference Laboratory on Alternative to Animal Testing, which has the responsibility to carry out a range of activities to promote the use of non-animal methods in legislation, biomedical science, and education. So the focus of my current activity is on the evaluation of innovative and integrated approach to assess chemical safety and support the regulatory implementation while also promoting 3 Rs across multiple regulatory sectors. I am also chairing the project platform of the European Partnership on Alternative to Animal Testing, and I act as contact point between the EMA 3 Rs Working Party and the GRC. So the aim of this session is to inform you on what is going on in the areas of 3 Rs, replacement, reduction, and refinement of animal testing for experimental use, and animal welfare more broadly. So we will give you an insight in how this activity aim at benefiting health and animal welfare. So we therefore will have 3 presentations. So the first one will be given by Dr. Sonia Beken, who will give a more general presentation on advancing acceptance of 3 Rs testing approaches for regulatory testing of medicinal products in the EU. And she will also introduce the newly established EMA working party on the 3 Rs. And then this will be followed by a presentation by Dr. Sarah Adler-Flint, who will go more into depth into the activity of the 3 Rs Working Party, which are related to veterinary medicinal products. And then we will move to the third presentation, and here we're happy to have Dr. Hans Spolder, who will talk about the recent development in livestock welfare science. So then after the 3 presentations, you will have the opportunities to ask questions and engage with the presenters. So I'd like to introduce, the first speaker, Dr. Sonia Beken, and she is the chair of the EMA Joint Committee for Veterinary and Human Medicinal Products 3 Rs Working Party. Sonia holds a Master in Biological Sciences and a PhD in Pharmaceutical Sciences from the Vrije University Brussels in Belgium, and a Master in Applied Toxicology from the University of Surrey. Sonia is also the coordinator of the unit of non-clinical evaluators at the Belgian Federal Agency for Medicines and Health Products, and her main areas of expertise relate to regulatory science, non-clinical drug development, toxicology, and metabolism, as well as alternative models to animal testing. So I hand it over to you, Sonia, for your presentation. Thank you.
Thanks a lot, Raffaella, for this very nice introduction, and thank you for having the opportunity today here to present the work of 3 Rs Working Party and to prepare a little bit for my co-chair, Sarah, and who will dig deeper into the veterinary topics. So I'm waiting now to have the slides on screen. Yeah, there they are. So next slide, please. So today I will be introducing to you different topics related to the activities of the 3 Rs Working Party. I'll first start to elaborate a little bit on the drivers for 3 Rs within the EMA, EMA's commitment to 3 Rs and the historical perspective to that, And of course, I'll take you to the heart of the topic of this presentation. It's to introduce the 3Rs working party to you. And it's quite ambitious 3Rs work plan. And then I'll wrap up with some take-home messages. Next slide, please. So first, for the drivers for 3Rs. Next slide. Yeah, thanks. So, of course, one of the first drivers is, of course, the animal use for experimental purposes in Europe. And if we look at the latest published numbers on that, we can see that about 7.9 million animals are used or have been used in 2020 in about 28 member states, which are the 27 EU states and Norway. And all these informations are, of course, available on the Allures statistical EU database. And next, please. If we look at human medicinal product use and the testing for medicinal products for human use, we can see that the majority of animals are being used to test for pharmacodynamics, including safety pharmacology in the topic of toxicity testing. And we encounter also a major amount of animals being used for quality control testing, and most of it for batch potency testing of human medicinal products. If we turn now to veterinary medicinal products, you can see a bit of the same picture, although most of the animals there in toxicity testing are used for target animal safety testing. But again, you see a huge amount of animals being used for batch potency testing, followed closely by batch safety testing purposes. So next slide, please. Of course, the use of animals for experimental purposes is regulated by Directive 2010/63, which we also have to comply with. And it's clear that Article 4 as well as Article 13 clearly enshrine the principle of the 3Rs being replacement, reduction, and refinement of animal use, and also with a clear preference for starting with non-animal methods and going further on in choosing a procedure to use the minimal number of animals, and also to apply the principles of refinement. Next. Although this directive is already quite advanced, in 2021, in September, the European Parliament agreed a resolution on plans and actions to accelerate transition to innovation without the use of animals in research, but also regulatory testing and education. Next. And in February of 2022, the Commission replied to this European Parliament resolution with different actions to answer to the call for acceleration of a transition to innovation without animal use. Next. And in this answer, we could see that the application of R&D, 3Rs in the R&D of medicines testing was put forward, and specifically there was a role for EMA and 3Rs fostering the 3Rs at the level of the European Medicines Agency. Next slide, please. Yes, thank you. Earlier this year, in May 2023, the European Parliament had a hearing regarding the European Citizens Initiative, Safe Cruelty-Free Cosmetics, Commit to Europe Without Animal Testing. Next, please. And the Commission again responded to this European Citizens Initiative with different actions. Next, please. And in this response, the European Commission committed to draft a roadmap to reduce animal testing as a prerequisite to transition towards an animal-free regulatory system. And these actions encompass the organization of multi-stakeholder workshops to kind of gear the work towards this roadmap and also discuss the state of advancement of it. Also, it was stated that there would be close collaboration again with the agencies. Of course, this involves the EMA again and the member state and relevant stakeholders. And there would be the analysis and the needs of the feasibility of an expert scientific committee that would provide advice on regulatory uptake of 3R methods, or the novel approach methodologies, as they are called also. All this, again, to promote the replacement, reduction, and refinement of animal use in regulatory testing. Next slide, please. So if we look at also the new evolutions regarding the reform of EU's human pharma legislation and the 3Rs, we can see that there are a lot of progressive statements in this new text for the directive and the regulation. And also already in the introductory considerations, there is, of course, the reference to the Directive 2010/63 and its 3Rs principles, which is logical. It was already there in the old legislation. But there is also the recommendation to avoid unnecessary duplication of animal testing through the installation of procedures that will facilitate joint animal testing wherever possible. But also there is a recommendation to reuse animal study results that are already available, and then, of course, to make animal study results publicly available, that as such that a reuse of animal study data will be made possible. There is also the reference to the one substance, one assessment work stream for environmental risk assessment. Next, please. And then in the articles, and there you will find also some very interesting statements with regards to 3Rs. The first of it is the request to demonstrate 3Rs compliance by marketing authorization applicants and holders. And it is just spelled out that the marketing authorization applicant shall demonstrate that the principle of 3Rs for animal testing for scientific purposes has been applied in compliance with the directive, and the applicant shall not carry out animal tests in case a scientifically satisfactory non-animal testing method is available. So this is quite advanced. Moreover, in the articles, a non-clinical test is more clearly defined, saying that it encompasses both in vitro, in silico, in chemical, and non-human in vivo testing. There is also the possibility to have the request to conduct a product-specific validation to replace an animal-based batch release testing as a condition for marketing authorization to really enforce the switch to a non-animal method. In the articles, there is also a large paragraph on the EMA responsibilities with regards to animal use and the 3Rs. And finally, again, the recommendation to have joint environmental risk assessment studies to avoid the unnecessary duplication of animal studies. Next slide, please. On top of all this regulatory background that enforces the 3Rs in animal testing, we are, of course, looking also at another driver for the implementation of the 3Rs, and this is the reduction of drug attrition through the implementation of better predictive models. Because let's face it, when we talk about 3Rs, we do not only talk about replacing, reducing, and refining animal use, we also talk about better science, better predictive models. If we look at the human space, we can see that there were already very early publications pointing towards reasons for drug attrition post-marketing authorization. And at the time, we could see that on top of the list were, in fact, lack of efficacy and emergence of new toxic events. Next, please. This review of 2004 was confirmed by later reviews by Harrison and by Hayes. And we could see that, again, lack of efficacy and lack of an emergence of new toxicity, were the main reasons for either failing of a drug during clinical trials or for withdrawal of the drug from the market post-marketing authorization. Next, please. And if we dig a little bit deeper into those reasons for withdrawal due to toxicity, we could see that most of the marketed drugs that were withdrawn from commercialization were due to emergence of liver toxicity, cardiovascular toxicity, and CNS effects. So a big reason of looking further at these endpoints and to develop better predictive models in that respect. Next, please. Let us move now further to the historical perspective on EMA's commitment to the 3 Rs. Next, please. EMA has historically been committed to the 3 Rs, and we can see this statement on the EMA position on the application of the 3 Rs in regulatory testing of human and vet medicinal products, and this dates back already from 2011. At that time, also a specific web page was set up on the ethical use of animals in medicines testing, where all information pertinent to the topic can be found. Next, please. At the same period, meaning in 2010, the first expert group on 3 Rs was set up, later on followed by the joint 3 Rs working group. And in fact, both groups really lay down the foundations for the work we can continue on today. So first of all, they set up a regulatory framework that will foster the uptake of 3R testing approaches in the European regulatory space for human and veterinary medicinal products. And I will just pick out one guideline that we drafted with this group, was the guideline on basic principles of regulatory acceptance of NAMs or 3Rs for the testing of human and veterinary drugs. And it really gives you the basic principles and criteria to comply with to bring a method towards regulatory acceptance. And several entry ports are also described in that guideline. Next, please. There was also an important part of the work that was dedicated to application of the 3Rs in batch release testing. You recall the numbers of animal use. A lot of them are used for batch potency testing and batch safety testing. So there is really a need to act in that space as well. In particular, we engaged into a review of final product batch testing requirements, and of course, on the basis of our findings, we made recommendations to marketing authorization holders to foster the use of 3R methods in line with the methods that are recommended in the Human Pharmacopoeia. And related to that, of course, we set up a training for assessors so that they would be updated about the new methods as well. A lot of the work of the JAC 3Rs and the joint 3Rs working group was also dedicated to collaborating with our international and EU stakeholders, being the European Commission, of course, EURL, EGVAM, and other EU agencies, and also EDQM. Next, please. So now for the heart of the topic, the new 3Rs working party. Next, please. So when we take a step back and we look at EMA's regulatory science strategy up to 2025, we can see two main core recommendations dedicated to the application of the 3Rs, and they are both for human as well as veterinary medicinal products. EMA wants to raise awareness for the 3Rs and NAMs and their regulatory acceptance, and for that we need really to engage into a discussion on the criteria for regulatory acceptance. On what basis can we accept certain methods in our regulatory framework? And therefore, we need to discuss qualification needs. What is the purpose for which a method will be used? So the context of use, what are the endpoints a method can predict, and what are the reference compounds you need to test your method with to show us that it really does what it says it will do. We also need to engage with stakeholders to do that, and for that we need to really build a European regulatory network on NAMs, and as you can see later, we are really moving forward in that respect as well. And in this science strategy, there is a focal role for a 3Rs working party. Next, please. So what is this 3Rs working party? It's a strategic and visible working party that will monitor and supervise all EMA's 3Rs activities. And to tackle this, because we are dealing with both human and veterinary medicinal products, going over a variety of disciplines, we are looking at quality control testing, non-clinical testing of human and vet drugs, and efficacy testing of vet drugs. We have also composition in a restricted core group that exemplifies this diversity and this multidisciplinarity. Next, please. Of course, we cannot do this alone with just 6 people. We have support of operational expert groups, and Sarah will discuss a bit about the OEG on batch release testing, and drafting groups, and of this newly set up non-clinical and new approach methodologies, European specialized expert community that will assist us and where we can exchange knowledge. Of course, we have the very dedicated support of the EMA Scientific Secretariat and the administrative one as well. And we have observers on board to close the loop. The European Commission is there, EGVAM is there, and ETQM. Next, please. So what's in our work plan? Next. As I said before, it's a very ambitious work plan with a clear vision to the future. And on a high level, we can see that the 3Rs Working Party really wants to put EMA in a strategic role in the field of 3Rs, with strengthened cooperation between all stakeholders and international partners. And I think this is really an important one, because we cannot do this just alone in our ivory tower as regulators. We really need to engage into a conversation with all involved to foster uptake of methods of in both testing of human and vet drugs. We really want to focus also on the transition of non-clinical assessment from discovery talks towards regulatory use and the acceptance of animal-free innovations of NAMs. Of course, we'll continue our work in the batch release testing space, and we'll follow up more on 3Rs for human and veterinary medicinal products. We will continue a review and an update of our guidelines to implement 3Rs best practices and do also some impact monitoring of the implemented changes. And then finally, of course, we'll follow up on all the actions that are recommended following this European Parliament resolution, and now also from the European Citizens Initiative work that was triggered. And finally, we'll also follow up and identify actions related to the alternatives to the use of non-human primates as well. Next, please. Just to give you a flavor on the guideline activities we'll engage for 2023, we'll draft a new reflection paper, again, on the alternatives to the use of non-human primates, and this will be in collaboration with our colleagues from the Non-Clinical Working Party for Human Medicines. We'll revise our guidelines on principles for regulatory acceptance. It has been out there since 2016. But we feel that it's time now to update it and include a definition of critical 3Rs related terminology. So what is qualification, what is context of use, and so on, to avoid confusion in the field and to have clear agreements on these terms. We'll include also specific annexes that will provide criteria for regulatory acceptance for organ-on-chip technologies, and this for specific contexts of use that will be applied in the pharmaceutical area. Finally, we'll revise our reflection papers that provide already an overview of the regulatory testing requirements and opportunities for 3Rs implementation, as was already stated once we rewrote them in 2015 and 16, that these would be dynamics overviews and will be updated as a function of technological progress. Next, please. So our work plan is really focused on regulatory acceptance of NAMs, and we have different actions that move us from the development of context of use-based qualification criteria, what are the criteria for acceptance of a method, up to the actual qualification of specific 3R methods or NAMs. Next, please. Yep. And we'll start off by organizing specific workshops, and this will be targeted for this year on organ-on-chip models. Next, please. We'll define regulatory acceptance criteria based upon the experience we gathered from those workshops. Next, please. We'll also engage into a conversation with our international regulatory partners, because you only can progress in the field if you have at least some kind of harmonization on agreement of what are these regulatory acceptance criteria. Next, please. We'll collaborate with our colleagues from the methodology domain because modeling and simulation is, of course, key if we're talking about in vitro, in vivo extrapolation, etc. Next. We'll support the qualification of NAMs and their 3Rs impact in various domains. One of them is embryo-fetal development testing, because there also a new regulatory guidance was drafted that has qualification criteria already embedded there. Next, please. And we'll also support, and finally, both the EMA Innovation Task Force and the Scientific Advice Procedure to assist in the actual qualification of methods that are submitted for qualification to the EMA. Next. Now, just a word on EMA's Innovation Task Force and the 3Rs, because that's really an important entry port for new method developers to come forward and engage into an early dialogue with the regulatory network. So it's a forum for early dialogue, and it's open to all kinds of stakeholders. And we're talking about small and medium-sized enterprises, academics, researchers, research and public-private funded consortia, pharmaceutical industry, and so on. The new focus that was published on the website is for regulatory acceptance of NAMs to replace the use of animals in the testing of medicines. So we're talking about in silico modeling, novel in vitro assays, and so on. And we really want to encourage the development of those and accelerate their uptake. It's an informal exchange of information, and from the network, you can expect some kind of high-level, non-legally binding guidance early in the development process, and it's really to gear you towards a possible qualification and possible regulatory acceptance of the methods. You'll always have the best available expertise from the network that will be sitting around the table, and the briefing meetings are free of charge. So it's really an open door to come forward and to discuss with us. Next, please. Of course, we'll have also some additional activities with the 3Rs Working Party, and they are listed here for 2023 and for beyond 2023. Next, please. And one of this I would like to talk to you today is the annual multi-stakeholder 3Rs Working Party meetings, of where we have started already with one in February of this year. Next, please. So to start the activities of the 3Rs Working Party and engage on a first time with our stakeholders, we invited industry and CROs, EU agencies and organizations, NGOs and animal welfare associations, research consortia, and other stakeholders to participate to a first stakeholder meeting with the 3Rs Working Party on the 28th of February 2023. We had a very busy agenda. We started with an introductory public session that was broadcasted live and was coupled to an interactive Slido to query really the audience on different topics. It was followed by closed sessions for every stakeholder group listed above, and the topics that we discussed were proposed by the individual stakeholders, and we had ample time for presentations and for Q&A. As an outcome, I can say that this was really a constructive day, and there were constructive discussions with all of the stakeholders, and the emphasis was clearly on the topics that were on the work plan. Of course, additional topics were considered, and they will be taken up for further action as seen fit. There is a public session report available on the website, and you can just scan the QR code to access it. Next, please. Just a few slides on the outcome of the Slido. One of the questions that we posed to the audience what do you think the most important aspect is when you think about 3Rs in regulatory testing and drug development? And there you can see that acceptance, validation, regulatory acceptance, collaboration, global acceptance are really buzzwords that pop up in the heart of this word cloud. Next, please. And then finally, also, we queried the audience for the main areas suggested for prioritization by 3R's Working Party. And it's clear that regulatory acceptance, qualification, and validation of NAMs or 3R methods is at the top of the list. And we really focus also our actions in that respect. Next, please. So let me now wrap up with some take-home messages. Next, please. Yes. So clearly, historically, the EU regulatory network has been open to the 3Rs. Next, please. You can go on. And EMA is clearly committed to the 3Rs. And this new 3Rs working party that started its activities in November 2022 is really EMA's official 3Rs hub. So the entry port for all 3Rs related questions with a 3Rs working party dedicated email address. We have a strategy for 3Rs and an ambitious work plan to support the work at hand. And it's really large and vast, and we'll not manage to do it all in 1 year. So we'll need several years for that, but we are really willing to move the field forward. Engagement and open dialogue with interested 3R stakeholders is at the heart of the whole action plan, and we really want to move together forward. Close collaboration, of course, with our colleagues from ITF 3Rs. It's an essential tool for early engagement, an open door to you to come forward with new methods. And finally, global regulatory collaboration is, to our opinion, also key, and we'll do everything which is in our power to make this happen as well. And with that, I would like to thank you for your attention and take any questions. And I'll now move over to my co-chair.
Thank you very much, Sonia, for this very comprehensive presentation. And I think it really sets well the basis also for the discussion we will have afterwards. So we'll have the question afterwards. So I would like to introduce to the next speaker. That is Sarah Adler-Flint, and she's the vice chair of the 3Rs Working Party. Sarah holds a PhD in cell biology and is a European registered toxicology. She's a scientific officer at the Department of Veterinary Drugs at the German Federal Office of Consumer Protection and Food Safety, and her main areas of expertise relate to stem cell biology, alternative methods to animal experiments, 3Rs, and regulatory risk assessment of plant protection products, biocides, and veterinary medicinal products. So now it's really a follow-up of the previous presentation, and Sarah will focus on the working topics related to veterinary medicinal products that are foreseen within the 3Rs Working Party. So Sarah, the floor is yours.
Yes, thank you very much, Rafaela, for the kind introduction. So after Sonia introduced you now to the very ambitious working plan of our 3Rs Working Party, I would like to focus now more on the topics that are related to the veterinary field. Next slide, please. So as you heard, the 3Rs Working Party is a joint working party of the Committee for Medicinal Products for Human Use and the Committee for Veterinary Medicinal Products. And it advises both committees on all animal welfare-related matters concerning the use of animals and the regulatory testing of human and veterinary medicinal products. So the working topics of the Working Party can be related to both human and veterinary medicinal products. So this is the most of our strategic work and the work on alternative methods or novel approach methodologies for non-clinical safety testing and overarching guidelines. But they can be also specific for either human or veterinary medicinal products. And this concerns the most of the guidelines, specific novel approach methodologies, or specific possibilities to employ the 3Rs. Next slide, please. So I will give you a short overview of the working topics that relate to human and veterinary topics, and it's a bit of repetition of what Sonia already said, but I will go shortly through this. Sonja already explained that we have annual 3Rs Working Party multi-stakeholder meetings for strengthening the dialogue and cooperation between all interested parties. We work on the creation of a worldwide community of regulators to harmonize regulatory acceptance criteria for 3R approaches and novel approach methodologies. We plan to provide training on 3R methods and best 3R practices across the EU network. And we strongly support the Innovation Task Force that Sonja spoke about, and also the Scientific Advice Working Party concerning 3R-related methods. Next slide, please. We review and update EMA guidelines to implement best practices regarding animal welfare. And Sonja already mentioned the guideline on the principles of regulatory acceptance of the 3Rs, replacement, reduction, refinement testing approaches, and that we want to include then 2 new annexes, one on regulatory acceptance criteria for microphysiological systems and organ-on-chip models, and another one on the definition of 3R-related terminology. And we plan to review the most promising 3R methodologies to be considered for replacing and reducing in vivo testing, and to establish a database for already qualified and validated alternative methods and novel approach methodologies, and this in collaboration with ECVAM in the EDICOM. Next slide, please. So now I come to the working topics that are mainly concerned to the veterinary field. So we work on the revision of the reflection paper, providing an overview of the current regulatory testing requirements for veterinary medicinal products and opportunities for the implementation of the 3Rs. We review VMP batch testing requirements with regards to the application of the 3Rs. We will contribute to a CVMP-CMDV working group exploring possibilities to make the adherence to 3R principles during authorization processes more transparent. And we want to include in vitro methods for local tolerance testing, skin irritation, and skin sensitization in the guideline on user safety of topically administered veterinary medicinal products, which is currently under revision. Next slide, please. So I come to the first topic, the revision of the veterinary reflection paper on regulatory testing requirements and opportunities for the 3Rs. The current version is from 2018, and the revision is currently in progress and is planned to be completed in 2024. The opportunities for the 3Rs, which are included, only concern successfully qualified 3R methods that serve decision-making for risk assessment of veterinary medicinal products in a regulatory context. And now also approaches for reduction and refinement have been included. And new is also a reference to the IDF procedure and scientific advice procedures in order to promote 3R methods. Next slide, please. So I will give you a few examples from this reflection paper concerning safety for pharmacodynamics and pharmacokinetics testing. There's now a sentence included that in vitro and in silico modeling can be considered if it's scientifically justified. For single dose toxicity testing, we have a statement included now on the questionable relevance for veterinary pharmaceuticals, especially for LD50 studies, and the recommendation to derive short-term toxicity values from other study types. Next slide, please. For repeated dose toxicity, the acceptance of one species on a case-by-case basis is included, and also the inclusion of additional in vivo endpoints in repeated dose toxicity studies, as for example, the integration of genotoxicity endpoints. And of course, this point comes also for the genotoxicity studies, so this endpoint can be incorporated into other in vivo tests. Next slide, please. For reproductive and developmental toxicity testing, the extended one generation reproductive toxicity study can be provided as an alternative to the standard multi-generation study. And for companion animals, reproductive toxicity studies are not expected for the evaluation of effects on the user, and standard developmental toxicity testing can be waived if significant user exposure is not expected. So this is, for example, the case for coated tablets, where you have a coating around the tablet, and so the user is not exposed to the active substance or other ingredients of the tablet. Next slide, please. For immunologicals, there are many new 3R approaches for batch potency testing included. So in many cases, in vitro methods are already preferred for routine batch potency testing. Then reduced number of controls are employed and batch potency testing for a number of vaccines and humane endpoints, which are specific symptoms that can be employed instead of the death as an endpoint, have been implemented. Next slide, please. So I come now to the second topic, the review of veterinary medicinal product batch release testing requirements with regards to the application of the 3Rs. So Sonia picked this topic or explained this topic already a bit. So we have the launch ongoing of the batch release testing operational expert group. So this is ongoing now in 2023. And the background for this is that for immunologicals and biologicals, each batch, which is a production unit, must be tested before being placed on the market. So these tests have to be done over and over again. And from 2011 to 2019, the precursor working groups to the current 3Rs working party reviewed batch release testing protocols of mainly centralized authorized veterinary medicinal products with a view to implement 3Rs compliant testing methods. And this newly launched batch release testing operational expert group is foreseen to complete and continue the above mentioned activity. And this will start with veterinary medicinal products, but it is planned that this will go on with human medicinal products in the future. So this operational expert group will include experts from the EMA network and the 3Rs working party with specific expertise in quality control and batch release testing, and also the 3Rs. And the goals of the operational expert group is the review of quality control and batch release testing protocols for centrally authorized veterinary medicinal products, and then later also the human medicinal products, with a focus on implementing the 3Rs. The group will make recommendations on implementing identified 3R compliant testing methods to the CVMP and then also the CHMP for the human products, and it will provide specific training for assessors. Next slide, please. So the third topic is the contribution to a CVMP-CMDV working group, considering how to increase transparency of compliance with 3Rs and marketing authorization assessment reports. And the CVMP plans to launch a working group to elaborate on an approach for making the adherence to 3Rs during authorization processes more transparent. This working group will consist of CVMP experts, including the veterinary 3Rs working party members and CMDV experts. And as the CVMP is responsible for all centralized procedures, and the CMDV, which is the coordination group for mutual recognition and decentralized procedures, and consists of all member states, and is responsible for all decentralized procedures, which concern procedures in which 2 or more member states of the European Union are involved. With this combination, we can elaborate on recommendations for all European authorization procedures. And the first meeting of this group will be in autumn this year. Next slide, please. This is on in vitro methods for local tolerance testing and the user safety guideline for topically applied veterinary medicinal products. And the guideline on user safety of topically administered veterinary medicinal products is currently under revision. And the 3Rs Working Party proposed the inclusion of OECD in vitro methods for local tolerance testing, skin irritation, and skin sensitization into this guideline. And this was endorsed by the Safety Working Party and the CVMP. And I will be also a member in the drafting group. Next slide, please. So the last 4 topics were topics that that are on the work plan now, but we also got some topics proposed from stakeholders. And Sonja already gave an overview of our 3Rs Working Party stakeholder meeting early this year in February. And one topic which was presented there was the guidance on exposure-based toxicity data requirements for veterinary medicinal products intended for companion animals. And to give a short background, data requirements differ between food-producing animals and companion animals. So for food-producing animals, which are, for example, cattle, sheep, pig, and chicken, and they derive meat, milk, eggs, we have to think of a risk assessment for the consumers and also target animals and users. And all the users are mostly professionals, so veterinarians and farmers. And then we have the companion animals, the pets, which are, for example, dogs and cats. And here we don't need a risk assessment for consumers, but only for the target animals and the users. And also the users differ, so these can be veterinarians, but mainly also pet holders or also the family members. So this includes also children and also pregnant women. So an example which was proposed was that according to legislation, the new veterinary medicinal product legislation for companion animal standard developmental toxicity testing can be waived if significant user exposure is not expected. And stakeholders strongly requested to have some more guidance on what is negligible exposure, in which cases these data could be waived. And this is a topic which will be discussed in the next year by the 3Rs Working Party, and we will decide then on further actions. Next slide, please. Another topic was, so there were more topics presented, but I have chosen to present these 2 because they are very interested for our working party to follow up. Another topic which was presented was at the CVMP Interested Parties meeting in May this year, and this was on phasing out the LD50 testing for veterinary medicinal products. And as I said already, some statements are already included in the draft of the reflection paper on regulatory testing requirements and opportunities for 3Rs implementation. And these are on the lack of relevance of LD50 studies for consumer and user risk assessment, and the recommendation to derive short-term toxicity values from other study types. Then for product-specific toxin testing for batch release, that is a topic which will be addressed in the future by the new batch release testing operational expert group that we presented. And concerning this topic, also the possibility of further actions will be discussed by the Working Party in the next year. And I just want to emphasize again that we are very happy to have this dialogue with the stakeholders, and that they provided a number of proposals, working proposals for us, and we are very happy to integrate these also in our working plan. Next slide, please. So this is my last slide already. Take-home messages. The EMA is clearly committed to promote animal welfare and the 3Rs. The 3Rs Working Party is the EMA's official 3Rs focal point. We work on a very ambitious work plan with focus on human and veterinary medicinal products. We have several specific projects for veterinary medicinal products in place. And these activities aim to promote the implementation of the 3Rs, for example, the development and update of guidelines and guidance documents, participation in the 3 Rs related working and drafting groups and other things. And as Sonja said, we are very much open with dialogue with interested stakeholders. This is also for us a very important opportunity. And at last, the 3 Rs Working Party contributes to 3R related innovation task force and scientific advice procedures. Yes, and with this, I end my presentation and thank you very much for your attention.
Thank you very much, Sarah. And I think it's good to see that there are already opportunities in place for the implementation of 3Rs. And I think it was also good for the audience to get some concrete examples of the kind of project that the Working Party will follow up. So far we have focused on work aimed at advancing the 3Rs in the regulatory context, but IMA interest in the field of animal welfare is much broader and covers different angles. So in recent years, there has been a high boost of activities in the field of animal welfare broadly, and we will get a flavor about recent development in livestock welfare science in the next presentation by Dr. Hans Spulder. And Hans is a senior scientist and EU account manager at the Wageningen Livestock Research, which is part of the Wageningen University and research in the Netherlands. He is an applied ethologist specialized in issues related to pig welfare. And between July 2013 and 2019, Hans was the president of the Health and Welfare Commission of the European Federation of Animal Science, and he is currently vice president for science of the same federation. Since 2012, he is a member of the Animal Health and Welfare Panel of the European Food Safety Authority. And in March 2018, the Commission, European Commission has designated the first EU reference center for animal welfare, of which he became the director. He is also a member of the management team of the EU Partnership on Animal Health and Welfare, and I think we will hear a lot about these different initiatives and activities in the next presentation. So Hans, the floor is yours.
Thank you very much, Rafaela, for this kind introduction. And thank you to IMA for inviting me to give you my bird's eye view on animal welfare, or at least livestock welfare science, with looking back at where we are, where we came from in terms of the societal debate on animal welfare, then a few slides on what is the type of work that we're currently doing in the scientific arena regarding animal welfare. And I will conclude with a couple of major developments that we can all see coming in the coming years. So my name was already mentioned. I work at Wageningen University and Research, and this is our wonderful campus, where we're all very close together from different scientific disciplines, not just animal sciences, also human sciences, plant sciences. And I think that's really important because, and that will also shine through hopefully in my presentation, it's important that we try and find each other and that we solve the big issues that we are all working on together and not in isolation in our own discipline. Let me take you back to through a brief history of animal welfare. This sounds very ambitious, but I'll keep it very short. In Western Europe, animal welfare debate, the discussion in society started about 200 years ago. This might actually be longer ago than some of you might think. In 1822, Richard Martin, parliamentarian in the UK, proposed the Cruel Treatment of Cattle Act in the UK. Martin was also the co-initiator of the world's first animal welfare organization, the Society for the Prevention of Cruelty to Animals, the SPCA. And that was established in 1824. And a few years later, well, 16 years later, Queen Victoria gave a blessing, and then it became the Royal Society for the Prevention of Cruelty to Animals. And I'm sure many of you will recognize these initials. The RSPCA is now therefore nearly 200 years old. This started the debate, but it mainly concentrated it at the legislative side. And in countries like my own here in the Netherlands, but also Ireland and some other countries, rules and regulations were very slowly established in the area of animal welfare, actually mainly to protect the humans from having a view on suffering of animals that may not be very pleasing to them. This sounds a bit cynical, but those were the first arguments for enforcing animal welfare legislation. Now, the thinking has changed dramatically, as you know, and a big push was given after the Second World War when Ruth Harrison wrote her book Animal Machines in response to the industrialization of livestock production. This book triggered a lot of discussion. It also triggered governments to think again about the way that we deal with animals. And the Bramble report, which was commissioned by the UK government, highlighted for the very first time a sort of definition of what animal welfare then should be. And Bramble proposed the 5 freedoms. And I'm sure this is something that most of you will recognize. The freedom from hunger or thirst, freedom from discomfort, the freedom from pain, injury, disease, freedom from fear and distress, and of course the freedom to express normal species-specific behavior. Very important, and guidelines which are still leading us in the discussion, although they haven't stood still. Things are moving forward in the area of animal welfare. And only a few years ago, David Meller and his team proposed a very important additional dimension to the 5 freedoms. And we now call it the 5 domains theory, whereby you'll recognize nutrition, physical environment, health, and behavioral interaction as important features. But what's now been added is the mental state, or the affective state, if you want to be slightly more scientific, of the animal. And together, these domains give a better definition of what animal welfare is and what we should be looking out for. There's another important element here, and that is that David proposed that we should no longer focus on reducing suffering, which of course is still very important in the current way in which we work with animals, but that we should try and take it one step further and try and work towards a life worth living for animals. And this, of course, is a notion, an idea that has far-reaching consequences on how we deal with our sentient beings, with the animals. Having a definition is one thing. The next thing is then how do you measure that? Because if you want to make progress, you need to know where you are and where you want to go, and whether you've achieved it. And in the area of animal welfare science, on the top right-hand corner, you see the old-fashioned way. When I did my PhD, I was still recording, even on a piece of paper, the behavior of the animals. Nowadays, things get automated much more, and the students type it directly into their iPad or onto their own computer. But behavior observation is an important element of assessing animal welfare. Bottom right, some more invasive techniques, in this case, the implantation of sensors to measure blood pressure. That technology has reached much greater levels, or very great levels in recent years, where sensors become smaller, become more accurate in helping us to understand the physiology of animals in response to the ways in which they are treated. Ethology, physiology, 2 important components. And more you see that these technologies are being automated. Artificial intelligence is used, machine learning is used to better understand what we see when we look at animals, and to try and translate that back to our management of the animals. These are issues mainly related to areas of behavior and physiology, but what do we then do with cognition? What do we do with the affective state? And this is not an easy one to resolve, but there is some thinking in the animal science domain on how we could do that, how to assess emotional state, how to know whether a pig or any other animal assumes a glass is half empty, or whether it thinks that the world is great and the glass is half full in this case. And there you need some ingenuity as to how you can ask that question. I'll give you one example, but there are more examples. Cognitive bias research started about 15 years ago, I guess, in livestock science. And cognitive bias research aims at understanding the risk-taking ability or willingness of an animal. Will it gamble that things go right, or will it expect things to go wrong? And that says something about the affective state of the animal. And the way it works, I'm not going in details, but just to give you an impression, the way it works is that you train a pig. I like pigs, so my examples are mainly from pigs. You train a pig to know that when it goes to the left bucket, it will find a food reward. Instead, if it goes to the right bucket, it will get an unpleasant, not a reward, but an unpleasant signal, a loud noise or a blow of cold wind or something like that, something that it doesn't like to do. Okay, you train a lot of animals to recognize this. Then what you do is you put them in conditions where you expect that they will affect their affective state. So you put them either in an environment where everything is fine, lots of toys to play with, lots of space to behave, versus an environment where that's not the case and they have more problems to cope. They don't like it, or at least you assume that they don't like it. And then you ask this trained animal the following question: what do you think of this bucket? Are you going to try and find food because you think there will be food there? Or do you think, nah, better not, because I've got some bad experiences in the past and it may not work this time and the world is all bad anyway? This sounds very anthropomorphic, but it works. And it doesn't just work in pigs, it works in quite a few other species as well. Even fish have been trained in this way and have shown differences in response, differences in choice, depending on the way in which they are kept. And now for something completely different. I was talking earlier about working together to understand animal welfare. And the first thing I want to state there is a notion which I come across quite often. That's that people are saying that animal health and animal welfare are the same, exactly the same. Now, I hope I'm preaching to the converted, but this is not my opinion. I think the two are different. To illustrate that, a short video clip of a group of sows, which to my mind are from a veterinary perspective completely healthy. They eat well, they reproduce well, they have a good body condition. If you look at the behavior of the animals, there's obviously something wrong. This is not normal behavior of a sow, and it's a coping mechanism to deal with an unfavorable welfare situation. And if this example doesn't convince you, then I have another one. These pigs are probably all, from a veterinary point of view, completely healthy and grow very well, but I'm sure you all agree with me that their welfare situation is, let's say, not optimal. So they are different. Animal health research, animal health specialists have a different angle of looking at animal welfare than animal welfare specialists have themselves, and we need to work together. And in one recent project that I was asked to coordinate, we tried to do just that, to deal with a slightly bigger issue, a bigger issue that we're all dealing with, which is to try and reduce antimicrobial resistance through reduction of the use of antimicrobials. And our project starts from the assumption that there are four strategies that you can use for that. First strategy is you have to stop pathogens from arriving on the farm. Biosecurity is key. If you don't have a pathogen, you can't get diseased animals. The second one is that if a pathogen should be on a farm, you want your animals to be as resilient or robust as possible so that they can withstand any negative disease consequences. Should they become ill, then the best thing is to detect that as early as possible. You detect that, there's many different ways. You look at the behavior of the animals and you very quickly see when a pig withdraws itself from the group or a chicken. So there are ways of early detection of diseased animals, and we should employ that as a third strategy. And then the final strategy, of course, is precision medication. Only medicate those animals that are actually in need of treatment. Now, together, these four strategies should reduce the use of antimicrobials, and they also show a clear collaboration between, on the one hand, health-related issues, veterinary perspective, biosecurity, precision medication, but on the other side, an understanding of the animal, a way that the preferences that animals have, the welfare of the animal in issues like resilience, development, and early detection. And just to illustrate resilience a little bit, give you an example of work by my colleague Ingrid van Dixhoorn a few years ago, who shows that housing, the way in which animals are housed, will actually promote resilience or be negative for the resilience of animals. What she did, relatively simple experiment, is to put pigs either in an enriched housing situation. Excuse me, before and after weaning, or in a barren environment. And then she imposed disease challenges, mild disease challenges on the animals, a viral infection with PRSV, and she also applied APP, which is a bacterial lung infection. And then she looked at how quickly these animals recover from this challenge. And what she found is that for the viral challenge that they got, that the enriched animals have a much faster return in terms of have a much higher viral clearance compared to the pigs that were kept in a barren situation. And for the bacterial infection, she found that pigs with lung lesions, pigs in a barren environment, have a significantly higher level of lung lesions than the pigs that were kept in an enriched environment. Now, the only difference between these animals were their ability to either play, explore, engage with other animals in one situation, but not in another. But it has an effect on how they deal with disease challenges. That was the last what I want to say on current research that we're doing. I have two, I think, quite important developments that are coming up, and that will be the last of my contribution. First of all, there is new welfare legislation regarding livestock species coming up. And just to give you the background to that, we just heard about the ECI on animal experimentation. A few years before, there was an ECI kicked off or coordinated, maybe I should say, by Compassion in World Farming, but many animal welfare organizations took part to end the cage age. European Citizens Initiative, in which our citizens in Europe were asked to agree with the statements. I don't know why I'm losing it now. Again, yeah. Asked to agree with the statement that animals are kept in cages, that cages inflict a lot of suffering. This is cruel, this is unnecessary, and therefore they asked the Commission to put an end to cage housing for, and then there's a whole list of animal species which are currently still kept to some degree, or to a large degree sometimes, in individual cages. In a very short space of time, they raised 1.5 million signatures, and therefore, just like for the other ECI that we just spoke, the Commission has to act and has to do something. And the way the Commission responded is that in the European Green Deal, in the section on farm to fork strategy, Frans Timmermans, who's in charge of or was in charge of the Green Deal, he's back in the Netherlands now since very recently, included two items on animal welfare. The first one was an evaluation and a revision of the existing animal welfare legislation, with proposals coming out now, Q4, well, nearly Q4 of 2023. And the second thing that was put in the farm to fork strategy is an investigation into better labeling of products in relation to animal welfare. Now, I put it in brackets. I'm not going to say anything about that, but if you like, maybe in a discussion we can talk a little bit about it. Let me focus on the legislation just by a few slides. The basis of the protection of animals kept for farming purposes stems from already 25 years ago. In 1998, this legislation was adopted. Since then, new articles, new regulations and directives have been released. And the most recent one is actually the one on the protection of animals at the time of killing. And you see that was in 2009. So it is a few years ago. Now, some might say that in Europe we have a very good animal welfare legislation, and really there is no need to consider whether we need any new. Others might say, okay, but it's been a fair few years since we've revisited it, and of course there is a lot of new scientific knowledge. Why don't we reevaluate? Well, the Commission decided to reevaluate, also to look at things that are not covered in the legislation yet. There's nothing on rabbits, as far as I know, other than a very general rules for animals, whether you agree with the keeping of them or not. Sheep, and even, to surprise of many people, nothing on dairy cows in terms of specifics for animal welfare legislation. And I could So something about foie gras feeding, about bullfighting, about things like slaughtering animals for religious purposes, so without stunning. These things have to some degree been regulated, of course, but it's good every now and then to revisit what we've done and how we agree with each other what we should do on animal welfare. So what the Commission does is they commission EFSA, of course, the European Food Safety Authority, to investigate the risk to animal welfare of the husbandry conditions that are currently in place. And the EFSA panel took this very seriously, and we produced something like 10 opinions for different species and different transport situations. And EFSA recommended quite far-reaching gave quite far-reaching advice regarding, for example, space allowance of animals, free farrowing systems for sows, transport durations, environmental enrichment, field calf housing. Anyway, you can read the list, and the list is much longer than this. You have to note that EFSA was asked to look at it from the animal perspective. We've been criticized a lot for not making realistic recommendations, and that is because still needs other considerations to be taken into place before you can formulate legislation. Animal point of view is one point of view, and it's the point of view that we were asked to reflect. So the opinions were then followed by an impact assessment in many different ways, organized by the EC with the use of various bodies, organizations over the past 1.5 years or so. And as I earlier said, there will be proposed legislative changes the end of this year. Thought it was September, but it's probably October, November before that will come out. After that, there'll be a period of member state consultation. And the idea, as far as I've heard, is still that in 2027, the new legislation should come into force. This is one thing I wanted to share with you. The other thing I wanted to share with you as a final one is very interesting for policymakers who need to know how to move forward. And it's very interesting for scientists because it gives us the opportunity to investigate some of the things that really have been lacking or are still missing. And what we would like to do, or the opportunity that we are going to get, is to do that through the European Union Partnership on Animal Health and Welfare. And Rafaela already referred to that very briefly. We wrote a proposal in response to the Commission's invitation, submitted it in April 2023, and we've now been invited for grant preparation, which in effect means that it will start, but it's just a matter of how will it start and how will it work exactly. And it should start indeed on the 1st of January next year. 56 research performing organizations, universities, research centers, etc. 30 funding organizations, so organizations from different European member states, and others, by the way, who will put money into this enterprise from, in total, 24 EU and non-EU countries. That's what the partnership is in terms of partner numbers. We've applied for 120 million euros to be spent on animal welfare and animal health science for the coming 5 years. The total budget can be much greater if the Commission agrees to the way we do it. If it's successful, if it serves a purpose, we might be asked to extend it to 10 years with a total budget of 360 million euros. The activities will mainly be two types. One will be internal activities by the partners of the partnership in project teams, and we will pay 50% from other sources. So the Commission pays half, the partners of the partnership pay the other half. There will also be external research calls where other parties can be involved, be drafted in to do research together with us. And these external research calls are paid fully by the funding organizations that I mentioned in my previous slide. So this is the basic structure of the partnership. EMA is an associate partner to the partnership, advising us mainly also on the links, of course. With their area of work. EFSA, the European Food Safety Authority, is also a partner in the project. My last slide, we start with a list of 17 internal activities. These have already been more or less agreed, and they include things like the surveillance of antimicrobial resistance, tools for the diagnosis of infectious diseases, positive animal welfare. We look at slaughter and killing for the assessment of consciousness and death, animal resilience that I just talked about, and also new antimicrobial and antiparasitic drugs and tools for efficacy and safety. testing. So these are 17 that we will start with. Each of these is about 5 million euros. So these are in themselves already huge enterprises. There will be 3 more internal calls and there will be 4 external calls during the lifetime of this project if we are successful and are asked to continue for 10 years. I'll conclude with a brief summary and take-home messages. The only thing that stays is change, and times are changing. We are slowly moving from reducing suffering towards a life worth living. Some will say too fast, others will say we should speed this up, but we are heading in that direction. New welfare legislation will be proposed soon. Welfare labeling of products, I'm sorry I didn't talk about that, but will increase in the EU, is my expectation. Animal health and welfare experts still have a lot to learn from each other, and we still should work more with each other, as with other scientific disciplines. And finally, there are great opportunities for policymakers in the coming years to put their questions to scientists and try and resolve some of the remaining issues, and for animal welfare and animal health scientists to work together across Europe in this huge big enterprise called the Partnership on Animal Health and Welfare. And there I end my presentation. Give it back to you, Rafaela. Thank you very much.
Thank you very much, Hans, for this very, very informative overview of activities and new initiatives in this area, both, I mean, with regulatory and also research angles and welfare, of course. So now I would like to open the floor for discussion and any questions or comments, questions to the different speakers. And comments are welcome. So maybe, okay, I see already a question here in the room. Please go ahead.
Thank you.
Could you please introduce yourself?
Yes. Hi, Afka Huizinga. I'm from DECRA. And I have a question for Hans. It was a very interesting presentation you held, and I fully support everything you've shared. I was just wondering how the farmers are being taken into account in this, the planning and preparation for this transition we need to make. Because, well, we all know farmers are not the happiest people at the moment because of all the rules that are already enforced on them. Yeah, are they being taken into account, or love to hear your vision?
Good question, of course, and thank you very much for that. It won't work if the farming industry, the farming sector cannot deal with it. The only thing that will happen if in the Netherlands, for example, we go too fast is that we export the problems. So the Dutch farmers will stop farming, and the work and the production of pig meat, milk, eggs will be placed, will be moved to another part of the world where farmers have a different situation that they can deal with. So it's crucial that in the transition, well, actually there should be 2 things. One is there should be people, organizations who keep pushing and who keep putting this dot on the horizon and say, this is where we want to be in the end. That's more like revolution. But I think at the same time, there needs to be evolution. And evolution is only going to succeed if you take all stakeholders on board right from the word go. So in the planning of what you want to do. So the discussions that we now have in the Netherlands on dierwaardige veehouderij, so an animal, how do you translate it? Animal-oriented or an animal-centered livestock husbandry, they need to involve, and they will only succeed if the industry is in agreement that there is a dot on the horizon that ultimately is achievable. So the initiation, the testing of new solutions, what we do in our scientific work needs to be done with stakeholders. And ultimately, when the legislations are formulated, when knowledge is being transferred, farmers again should be on the forefront, but also veterinarians, they play a crucial role, should be at the forefront of receiving and of the dialogue. Sorry, I'm taking too long with my answers.
No, that's fine, because you also responded to another comment that was in the chat, which I will read. Difficult to implement. You said really interesting what you said on animal welfare and your views. Difficult to implement with some of the farmers we have in EU. but we'll keep on insisting and working to make this view global. The higher the welfare standard, the higher the revenue. And there is a follow-up question. Is the EU going to ask for the same standards for importing products? It is a concern for farmers, at least that is what they say and ask.
Yeah, so it's good to know what you do and where you are. I'm not working for the Commission, so I cannot answer that. I can only answer what I hear or what I believe that should be done. And I think, just like what I just said, over exporting the problem, if we do not ask for a similar level of animal welfare for products that we import, then of course we will be exporting the problem. But again, I'm the wrong person to ask what the Commission is going to do. That's up to Brussels.
Clearly, the global dimension is always really key. There is another question in the room.
Yep, thank you very much. And Dr. Pulder, thank you very much for this very interesting presentation. And yeah, as you know, animal welfare was my first love, so it brings good memories. Two questions. One is, do you know this new legislative act that is coming is going to be a regulation? Because as you might know, the problem with all these directives, not only it's the date, but it's also the fact that they've been implemented quite differently in the different member states. And the second question is, in the slide with all the projects that are going to be funded, there was also a point, if we are not wrong, about a vaccine platform. So if you know anything more about it that you can share with us, exactly what that project will focus, if you know, otherwise it's, thank you very much.
To start with the second, no, I don't know the details of the vaccine platform, but what I understand from it is that there's quite a large number of organizations linked to the partnership who are involved in vaccine development, and they would like to join forces to unduplicate and to be complementary to each other. And for that, they've set up a platform. That's one of the initiatives of the partnership. No, I don't think the details are public, so I can't even share it with you at the moment. We have to wait until the project is granted, and then the details will become available. Your first question was a more tricky one.
Whether if the new legislation that you mentioned on animal welfare,
Right.
it's going to be a regulation.
I don't know.
Okay.
I don't know. I don't even know whether it's known.
Okay,
from the
then
outside
no
world.
problem. Okay.
EFSA gave very specific recommendations.
Okay.
So in that sense, the legislation could be fairly detailed, but of course it's up to the Commission how they are going to frame it.
Thank you.
Hi, I'm Carl from Health for Animals. So I have a question to the first two speakers on the three R's. So I work for a global association of pharmaceutical producers, veterinary pharmaceutical producers, and I was very happy to hear that both of you mentioned the importance of global regulatory collaboration on this subject. I wrote down the worldwide cluster of regulators. I don't know what that is, but could you expand a bit on that? Because as you've both kind of alluded to, and also I think from the stakeholders' feedback, the global cooperation on this subject is very important. And I say that because as a representing manufacturers, we develop products not for one country in Europe or for Europe alone. We develop more and more products for global markets, and particularly 2 or 3 other markets are very important to have along with you as you move forward. So I'd love your views on that. Thank you.
Yeah, thank you for the question. It's indeed a very important point, and it's not new. That's why we have platforms such as ICH and VICH to at least harmonize requirements across the main players, being the United States, Japan and Europe, and with accession by Canada and Switzerland and other regions of the world. But to go even faster, we need this informal dialogue on an international level. And it's already happening now, very ad hoc on certain topics, but we felt that there was a need to have this more strengthened to prepare also the work in ICH and VICH that, as you know, might take a lot of time before changes are implemented. So as such, we reached out to our sister agencies around the globe and to engage into this dialogue. And we are slowly moving forward, I would say, but I think there is an interest from both sides of the pond, but we need to set up the exact terms under which we could engage into such a dialogue. But there is certainly interest, and conversations are ongoing, like with FDA and with Japan, more very informally on this topic. But it will focus on a first instance on the qualification criteria, because that's where there are lots of discrepancies. And if we can already have agreements there, we could foster uptake more swiftly later on in ICH and VICH. I don't know, Sarah, if you want to add anything.
No.
Okay.
Can I just follow up on this question? So will this also involve agencies or regulators from other countries, in addition to those that are already in ICH?
Let's start walking before we can run and do it slowly, and to get, we can of course discuss with other agencies, and I see that at conferences also other like the Korean colleagues are also coming to us and asking for our experience, and it's a kind of a learning exercise right now, and we'll see how it moves forward. Thanks.
Other questions in the room? Okay, yes, then we will move the questions online.
Sorry, I thought I have another question, and this one's for Mr. Spoelder, Dr. Spoelder, which is, you mentioned welfare labeling in your presentation. Do you mean labeling on consumer products of how the animal or the animal protein was raised, or whether it meets, is that what you meant, welfare labeling? Could you expand on that, please?
Yeah, apologies for not having been very clear on that, but that's indeed what I mean. So when you go to the supermarket and you decide what to buy, that you're able to see, to have an impression of how the animal was kept. So what their welfare conditions were. And of course we have that, for example, for eggs, where you can quite clearly see whether it was a caged egg, which should not happen anymore, of course, or whether it was free range or organic. In several European, mainly Western European countries, we have these schemes in place already. Supermarkets have already embraced them. Denmark with the 3 Hearts, Netherlands, Beter Leven, Hallmark, and there are several more right across Europe. And the Commission has set up a working group to discuss whether this should be made mandatory. So whether there should be compulsory product labeling reflecting the rearing conditions of the animals. I think the Commission in the end endorsed the fact that labeling increases, but have suggested that it should be on a voluntary basis. I think that's the long and the short of the discussion over the last few years regarding labeling. And we'll see where it's going to take us from here. It's an opportunity. Welfare is often branded, or whatever the word is, as this is going to cost us money. It's an opportunity to make money. And I know it will not be an opportunity for everybody, but at least for those entrepreneurs who are very keen and who are able and who are willing to take those steps forward, it is a way of making a little bit more on by keeping animals in the way that is more friendly for them than in many of the conventional ways that we have now.
Thank you, Hans. So since we have industry representatives very active in the room, can I ask a question? So we heard in the initial presentation that there is the human pharma legislation, upcoming new proposal, and there are several considerations for the 3 Rs, including avoidance of unnecessary duplication of animal testing, so procedures to facilitate joint animal testing where possible, or use animal studies results and public availability of animal study results. So I wanted, yes, if you could comment on that, and if you think this is really feasible. It is already done in other areas like industrial chemicals, for example.
I mean, let me be very clear about this. We welcome any reduction for multiple reasons, some more altruistic and some more humanitarian, or if that's the right word. So yes, we would welcome any form of, and we have actually put forward a number of different ideas in different entities around the world on how to reduce the number of animals used in testing, including, I think our initial focus was in batch test release. You probably know more about that than I do at this point. Yeah, we would certainly support any of those approaches. Again, most in the veterinary sector, most veterinary products are put on global markets, 5, 6, 10 markets. And so if we can have that coordination and that leads to a reduction, we would very much strongly welcome that. Again, we are there to obey the law, so we're happy if laws change and we benefit from that, but and the animals benefit from that, I should say. So I don't know if that's a proper answer to your question. If there are other colleagues who want to answer, respond to that.
Yes, I mean, this is a good answer, but I was meaning also about joint animal testing. Is this feasible between different companies?
I don't know. I would love to say I know the answer to that one. I don't know, but it's I can take that one home. Thank you.
Yes, we will go then to questions from. Okay, Michael.
Yeah, thank you very much for that response. Just a quick follow-up, actually, for the message to take home and ask. I would even go further, and not only joint testing, but also sharing of data, because that would also reduce animal testing if you share with other entities data on a certain compound that has been already done. So that, of course, will have an economic impact, but on the other hand, it will reduce animal testing. So is that an option, and do you consider something like that?
Yeah, I'll take it home.
Sarah?
Yes, it was also mentioned that in other regulatory areas, data sharing is already taking place. For example, in the pesticides and biocides regulation, and there the companies actually obliged to share the data, and with a letter of access, which the other company has to buy then. So there is a financial, how do you say, incentives. So that not one company has the whole burden of the financial burden, that the other company who buys the data actually of the other company, but there is the obligation for the company to share the data. Could you think of, what do you think of such an idea for the veterinary field? That would be very interesting.
To be honest, I don't know. I'm not familiar enough with that. Again, like as I said, I think we'd be happy to look at it and consider it, but I have not consulted with so I can't really answer that question. I'm sure those discussions have happened in one of the fora which our industries are part of, but I don't know the conclusions of that. So I would say that if there's a precedent in human pharma and other areas, there might be some interest there. I could also think of all kinds of other reasons that might be more prohibitive toward that approach. But again, if it works in other sectors, could work in ours. But I just can't answer whether. I just don't know enough about it right now. So,
Okay.
but we will forward you all our experts on this.
Okay, thank you a lot. A question here.
Yeah, no, just a comment, maybe to put it a little bit in perspective also. We do have the concept of generic applications, and that means that, an innovative product comes to the market, and of course that innovator company needs to do the testing. But after the data protection period has ceased, generic companies can make reference to the data. So they don't need to redo all the studies. They can just show that they are bioequivalent with the reference product, and therefore then they can rely on the data that is already there. So there's no duplication of data there. So I'm not sure what type of data sharing that we're talking about here. I think, for example, for batch release testing wouldn't be a solution because that's related to the particular batches of the company. So I don't think really data sharing is a very big point here in veterinary medicine regulation because we already have those concepts in place. Thank you.
Thank you very much for this clarification. So I think we'll maybe go to the questions online.
Yeah,
Michael.
thank you very much, Rafaela. So I'll read out the questions online that we received. So this one is one for Dr. Spulder. So it reads, you mentioned focus or initiating on AMR testing in veterinary pathogens. Could you expand a bit more on which specific pathogens this program will be focusing on, and i.e. why we also have national monitoring programs in place in several EU countries? So would it be an addition to already existing national surveillance programs?
Yeah, good question. No, but for two reasons. So I cannot elaborate because I'm not a specialist in the area. So I wouldn't be able to give a precise answer to the question related to specific pathogens. But the second part of my answer is more relevant, and that is that what we're doing here is we are testing principles. So we are using an infection with a pathogen as a model to challenge the animal. And it could probably be any other mild pathogen. To see whether the assumptions that we have, whether the hypotheses that we have are actually valid. So for those two reasons, no, I don't know much more about the specific.
Okay, thank you. Thank you very much. Okay, then the next question from Paul. This one is probably one for Sonia. So what is the relationship of the 3Rs EMA Working Party towards the European Partnership for Alternative Approaches to Animal Testing?
That's an easy one. We are involved in the steering committee and also in the project platform meeting. So we are liaised with the EPA, and we collaborate also frequently in their partner forum meetings where we engage with the other sectors to try and advance the field.
Thank you very much. Sorry. Yep.
Yes, I think, there is also a lot to learn across sectors, I think. And I was just wondering, how can networking be also enhanced across sector? Within the medical area, you have veterinary, human, different areas of medicinal products.
Yeah. I think with the 3Rs Working Party, that's what we are trying to do, because it's really, I think, the most multidisciplinary working party here in the EMA, if I'm not mistaken, and I'm looking at my EMA colleagues, because we have human vet on board, we have people that are involved with quality testing, non-clinical testing, and clinical testing. So it's a bit of everything. And we historically in the JAC 3Rs had this as well, and in the 3Rs working group. So it was really a learning experience, because also reviewing all the guidances that are out there, EMA guidances, ADQM monographs, as well as ICH and VICH guidances, learned us a lot. And already we took over some recommendations from the human side towards the vet, and inverse we learned from the veterinary side. So I think this is an amazing experience, and it's really a plus. Yeah.
Good. Very challenging as well, I suppose.
Thank you very much indeed. That can be quite challenging at times. Okay, so there's another question from Sam Saunders for Sonia or Sarah, which states that, would you support including a provision in the human medicines legislation to formalize the 3Rs WP and give it a legal mandate?
That's a tricky one. It's difficult to give an opinion on that, but we should also keep in mind the response of the European Commission to the European Citizens Initiative, where they mention a scientific committee on 3Rs, and maybe this is a kind of more formal type of 3Rs group that could be envisaged for regulatory testing of compounds. But that's all I really can say about this.
Yeah, thank you very much, Sonia. Indeed, this is a tricky one, and obviously it's the Commission's mandate to decide ultimately on this or not. Just to say that the entities within EMA that do indeed issue the opinions are the scientific committees, which are supported by the working parties. So formalization in that sense would then be required for all working parties, not only for the 3Rs working party, but formally it's the committees, that do the scientific work, which are then supported by the experts. So yeah, I think this is an issue that can be discussed, probably is discussed also with the Parliament proposal, but yeah, hard to give a final answer on that, I guess, at this stage. Yeah, thank you very much for that question. So we have one more, and I guess this is one also for Sarah or Sonia. Thank you for the presentations. Regarding the 3Rs working party, will specific measures, timelines be included to assess impact, success, i.e. reduction or elimination of numbers of animals? Thank you.
Yeah, we mentioned impact assessment in the work plan because this is something new that was not really included previously. And we would like to start with very specific topics related to ICH guidelines that were approved. For instance, the ICH guideline on reproductive toxicity testing, we really have qualification criteria endorsed on a global basis in there for alternative models. And there we can indeed try to keep track of the impact of this new legislation on uptake of new models and maybe use of models. To link this to the statistics is another problem because the statistics level of granularity is not so high to be able to monitor, plus studies are conducted around the globe. It's a global industry, so it could well be that reproductive tox studies are done in the United States or somewhere else for products that are marketed here. So we really will have to think carefully of how to measure impact of legislative or guidance changes with regards to 3Rs. But we're not there yet, but we are taking into account the complexity of this indeed.
Yeah, indeed, and if I may add, this is indeed a very complex question because if as you said, if you just don't, if you want to go really down to the core of it, you'd have to look at the data and the dossier submitted to really look at the numbers that were used. Just to let you know that these kind of data, of course, are usually confidential, so we can, of course, not release them. But in an aggregated form, we do every now and then in internal and isolated scientific projects look at the impact of certain measures on certain specific areas. But in the frame of the 3Rs working party, as Sonia just said, we would have to think about how to do that carefully and also consider the resource implications of this, because looking at many dossiers and actual numbers will be very resource heavy.
We did a kind of similar thinking exercise when we looked at the impact of the new guidance related to carcinogenicity testing of human pharmaceuticals with the weight of evidence approach that was implemented instead of asking immediately for a 2-year or 6-month carcinogenicity studies in mouse and rats. There is a weight of evidence to decide whether or not a long-term carcinogenicity study would add anything to the risk assessment. And in that context, we were thinking how many animals would be spared by the implementation of this new guidance. And it is extremely difficult because there are a lot of factors that come into play. When do you really need these carcinogenicity studies? What type of products, what type of modalities, what type of indications, and then the global market. So it's kind of speculative to think about this. And we really need to have kind of tools to measure this, or at least surrogate markers of impact.
Yeah, if I may add, yes, I mean, that's really tricky. And maybe it's something that is easier done retrospectively than just an estimation. I know that the European Chemical Agency is trying to put together this type of data. And I also wanted to mention that also there is work ongoing in the area of biomedical research to try to establish indicators to see really what is in the impact of the European research in the area of 3Rs.
Thank you very much. So the next one is for Dr. Spuler again, and I think very interesting actually. It's from Jos Olatz, who asks, welfare legislation is very much orientated on husbandry conditions. Is the angle of genetic diversity considered within the framework of animal welfare? And then he gives the example of the Belgian blue breed of cattle, which has to be always delivered by cesarean because the calves are so big that they cannot be delivered naturally. So, or increasing the fattening potential in poultry, for example. So are these, genetic factors, shall I say, or breeding factors considered?
I think breeding is a crucial element in improving animal welfare. On the one hand, breeding, of course, was used a lot when we started to professionalize, some would say industrialize our livestock keeping. Breeding then also had its negative side effects, of course, or breeding goals had their negative side effects. But what you see nowadays is that the European organization, or at least the European organizations that deal with animal breeding, have developed a much more sustainable strategy. They even got their own agreement, I forgot what the name now is, but for to develop sustainable breeding goals. And that also takes into account various aspects of animal welfare. Examples like litter sizes in pigs. If you have too many piglets, more will die. So can you formulate a breeding goal in a slightly different way that you have better survival rather than more piglets born? Those are the sorts of developments that are now ongoing in genetics research, and very important, I think.
Yeah, I fully agree. This is indeed a very important aspect of animal welfare in husbandry. Thank you. So there's another question from Adarja, which asks, concerning the promotion and facilitation of 3Rs and non-animal testing alternatives in, for example, batch release testing, how can we ensure that potential use of alternative methods does not impact the overall quality of data compared to current methods? So I guess this is one for Sarah. Thank you very much.
I'm not sure if I understood the question correct. So is it meant that we cannot cut down on safety issues for?
Well, I guess the question focuses on how can we ensure that alternative methods used within batch release testing will be as safe, and I put safe into, I don't know what you call these in English, but inverted commas, as the currently used methods, which are usually in vivo.
Yeah, so I'm not an expert for batch release testing, I have to say at first, but you can be very sure that we don't use any alternative methods, in vitro methods, if they are not proven to be safe, if they have not been qualified and validated. So if these methods are used, they we will never cut down on safety for patients, consumers, users. All alternative methods we will apply at the end will have to be proven to be valid. Thanks.
Yeah, maybe I can emphasize on this. It's the collaboration we have with the EDQM and with also EGVAM for a lot of these methods that are either validated through a collaborative efforts with EGVAM and EDQM, and must say that also EDQM is engaging in a lot of collaborative validation studies for all these 3R methods for batch safety and potency testing. And once proven valid, they are inserted in their monographs and stated as a valid method and listed as a method that you can use. Of course, to implement it, you will have to do, again, a product-specific validation, which can be lighter if you were involved in a collaborative study, and we have a guideline on that. And this will, again, be assessed by us in a variation dossier that's submitted to either the EMA or national competent authority if it's a non-centralized, centrally authorized product. So there we have, again, a check to see whether the method performs adequately according to the standards that are clearly stated. So that's the insurance that you're asking for. Thank you.
Thank you. So we heard about this new task force, innovation task force. So I think that's really very good tool for early engagement. And maybe I'll ask Michael, and if you could maybe elaborate a little bit on that.
Yes, thank you very much indeed. We would like to use this forum actually to maybe advertise this a little bit more. So the ITF, the Innovation Task Force, that now has been specifically open to 3R's method, is indeed intended for early engagement of method developers. And there we really want to stress it's not only pharmaceutical industry, it's also academics. It's basically everybody that has a method that can interact with the agency and all the 3R's experts of the agency to actually discuss already at an early stage, and I shall say free of charge. That's an important parameter here, because the scientific advice procedure that was also mentioned later is not free of charge, even though for certain stakeholders like small and medium enterprises or certain others, there is considerable reductions in fees. Nevertheless, there's still a fee associated, while for the ITF, it's a free of charge discussion where you can engage with the agency and actually discuss whether a certain method you have developed might be suitable or not suitable for regulatory recognition, and whether, or you could also get tips on what needs to be done in order to qualify this method, and whether this method is actually already ready for qualification. So you from there on, you would be advised on what steps to take in order to have a formal qualification of that method within the agency. And in that context, I also wanted to say that we use the term qualification rather than validation, I guess, here at the agency, because very often when we talk about acceptance or regulatory acceptance of alternative methods, there's a lot of terms that are used, and everybody or a lot of people think that the use or regulatory acceptance of an alternative method is always associated to a full validation through multiple laboratories, etc. And for, as Sonia said earlier, for specific contexts of use, actually, this is not the case. So the whole acceptance and use criteria for an alternative methods for a specific context of use is not as elaborate and complicated, shall I say, as one might think. So please come forward, start discussing with us. It's actually quite straightforward. It's just a one-page document that needs to be filled in with what you want to discuss with us, and then we'll do the rest. So please come forward and as much as possible. Thank you very much.
Sonia.
Yeah, maybe just an additional word on this context of use. It's a terminology that we use all the time, and I'm not sure if everybody understands it. It's very important for the method developers that are here in the room or that are online. It's really think, what's the purpose you want to use your method for? And in the regulatory context, and that can be quite diverse. You can be using your method for a demonstration of proof of concept of your drug, which can be veterinary or human, up to pharmacokinetic testing, quality control testing, non-clinical toxicity testing. So it's quite diverse, but you need to think upfront where you would see the method. And based upon this discussion, we can also give you really valuable advice in these ITF meetings of what would be the qualification needs, as Michael said, which can be quite diverse depending on this purpose of use. But that's also the thinking process that you need to do upfront before coming to this ITF meeting. And we can help you fine-tune this, but it's important that there is already some idea about possible application or context of use. Thanks.
I think it was really important to clarify this, and we will hear more about qualification because the working party is aiming at working on criteria for qualification for context of use. So we'll see the progress of that maybe in the next stakeholder meeting. I just would like to check if there is still a question in the room. Yes, Nick.
Thank you very much. A question for Dr. Spoolder, if I may. So you indicated that that there's clear evidence that the efficacy of a veterinary medicine will be impacted by the welfare conditions in which the animal is living. So do you have any reflections on how that should be considered when evaluating the efficacy of a medicinal product? I mean, clearly in a single clinical trial, you would expect the animals to be housed in the same way. But if you're comparing the results of different clinical trials, what does that mean? Should we be looking towards some standardization of welfare for clinical trials or something? Thank you.
Thank you very much. There are, of course, different elements or aspects that determine whether a disease gets hold or whatever, whether an animal becomes diseased after an infection. What I was talking about was increasing the intrinsic resilience of the animal, so its immune system as it's affected by, for example, stress levels in the animal. That's the primary, that's what I was talking about. Of course, there are other ways. Vaccines, other medication will also help. Well, we know that's the business that we're in here, but that will also help to combat disease, and that's another way. So I don't think resilience in that sense, well, it may affect efficacy testing actually if there is an interactive effect there. So if you, not the general status of an animal itself can be compared across the application of the medication or not, but whether there's an interaction, that the efficacy of the vaccination is actually dependent on whether the animal was kept in the good circumstances or whether it's kept in bad circumstances. So that's an interactive effect. I don't know, I have no evidence of that. I think there might be a cumulative effect, so that they each work independent but towards the same goal, but I'm not sure whether there is an independent effect, and therefore that you have to take into account what the husbandry conditions are when you test a vaccine, for example. But that's my layman interpretation of this.
Okay, so we see again the, interaction between the areas of health and welfare. That's something really probably that really needs to be enhanced, and where a lot can be learned. You wanted to say something?
Well, I was going to say that yes, but how these two interact, I'm not 100% sure. So whether they're simply additive or whether they are actually interacting with each other, I don't know. But they're, of course, both very important.
There's just one last question online, and I think this is really the very last question.
Yep, thank you very much. And this is from Jose, who asks, according to the labeling, there are different private standards in place that already permit labeling. How is the EU going to try and standardize the standards so they are the same level?
Level playing field. We haven't mentioned it here yet, but of course that's extremely important. At the same time, diversity is also important, and labeling creates the opportunity for diversity. And then you can either organize that across the whole of Europe through what we've done with the eggs, for example, or you can leave it up to industry to devise their own system of labeling and then take the advantage of doing so. Depends on your point of view, whether you think you should go one way or the other way. Both can be effective.
Okay, thank you very much. So it's 11, so I think it's time for coffee, and I really would like to thank a lot the presenters for the great presentations, and also we saw that this really triggered a very good discussion with really diverse type of questions. We had also diverse topics. And so I hope you really enjoyed the session, and thanks a lot very much, and also Michael for the support.